Ma'am, I think I will also join in multiple devices or switch at some point.
No problem. Sabarna, the participants are joining. We can start at 4:35, right? 4:35
4:30.
dot we will start. All right.
Okay, okay. Yeah, yeah. Okay. So that means, yeah, okay.
So I have means communicated to the participants regarding downloading of R. So might be they have downloaded it.
Okay. Okay.
Sabarna, I think we should start, right?
Okay.
Um, good afternoon, would be Dr. Sabarna, and good evening to our participants. Today, I'm really very happy to introduce you to our own student, going to be doctor very soon, Sabarna Bhattacharya. And Sabarna did his BSc Honors Botany from our own college, that's Zakir Hussain Delhi College, University of Delhi, in the year 2018. Then he did his masters and currently on verge of completing his PhD from University of Bonn. And currently he is research associate in the same university with Dr. Watt. I'm sorry if I pronounce it wrong. Dr. Watt Netchif, Department of Plant Cell Biology, IZMB, University of Bonn. Dr. Sabarna is also awarded with several accolades, awards, and achievements. He got certificate of merit and merit scholarship from Zakir Hussain Delhi College in the year 2016. He was a university rank holder, meritorious scholarship award he got from Faculty of Science, University of Delhi in the year 2016 as well as in year 2017. And also he is awarded with TRA 6 Research Booster Grant from University of Bonn. Dr. Sabarna is also have research articles, several research articles in Journal of Repute, International Journal of Repute, and also have in our college also have given several lectures, hands-on sessions on computational skill as well as transcriptomics and so many other techniques. And also he is proficient with data processing in R, MATLAB, RNA-seq and WGS, data analysis, machine learning, SVM, clustering techniques, and so on. With this introduction, I would like to welcome our own Sabarna to kindly deliver his session. Thank you, Sabarna. Thank you. Being like we have time gap between Germany and India. So thank you very much for sparing your time for us. Over to you.
Thank you, ma'am, very much for the nice words of introduction. And it's honestly a pleasure to get associated with this wonderful faculty at the Zakir Hussain Delhi College, and it has always been my pleasure. So maybe I will already start and start sharing my screen. So. Okay, ma'am, I think I am not able to share.
Let me make you a co-host. Just a moment, please. Please try now, Sabarna. Are you able to share now?
Yes, just a second. I think so.
Yes, your screen is visible now.
Yes, okay.
You can just make it full screen.
Yes. Can you see me now?
Yes, I can see you, but it's not yet full screen. Like after sharing, you share your screen, the entire screen, and then make it full screen.
Okay. Let's see if it works here. Now, can you see it?
Screen is visible, Sabarna, but the problem is it is not full screen. We can see your side panels as well.
This is strange.
Have you shared entire screen and made the presentation full screen?
Yes, but I can try it again.
Instead of sharing the windows, please share the entire
screen.
Then only there is some problem, I guess. So you'll be able to do it full screen.
Okay. Entire screen. Wait a second. Can you see me now?
Yes, but it is still not full screen. the
Okay.
presentation.
Now?
Yes, yes, yes.
Okay.
Sabarna, one more thing, please. Just click on this hide as well, because sometimes it hides the thing that are there on the screen.
Okay. Okay.
It's perfectly fine now.
Okay. Yeah.
If you want, due to connectivity issue, you can switch off your video as per your convenience. No problem.
Okay, madam. Yeah, yeah, yeah. I will see. Just a minute. Uh, mm, yeah. Okay, let's get started.
All well,
Okay.
Sabarna. Very
Yeah.
fine.
Yeah. Okay. So, hello to one and all present in this meeting today. And as ma'am explained nicely, one of my kind of interests has always been in transcriptomics and analysis of big data. So it's so wonderful to be here in this meeting today. So since it is going to be interactive, I decided that maybe we can do it in 3 parts. And in the first part, I will share certain theoretical information very shortly so that we know all of us are in the same level of the analysis that I'm going to show, including backgrounds and certain stuff about RNA itself and RNA-seq analysis. And as you know that I will try to do this session today in 2 parts, and later on I will rejoin from a Linux computer to show you the alignment steps and the preliminary steps that are involved. Once we are done with the Linux part, I will show certain examples and how to proceed with that. And once we are done, we can do a short question answer session, and then we can do a short break. And after the break, we will do differential expression analysis that will be done in RStudio. And I think many of you have already installed RStudio and have it on your computer as well, so it should not be a problem. And with RStudio, at a certain step, I think there we can do a really interactive session, and we can do the differential expression analysis together. And that might include, and that might involve generating graphs and seeing some data. And also we will try to do a technique called gene ontology, which I will explain to you as we come closer. So I think that would be the game plan for today's talk. And yeah, so firstly, we all know, I think there are also students here, that the central dogma of biology says that how information flows. So it's from DNA to RNA to proteins. And in the past decade or so, there has been great advancement in sequencing technologies, something known as next generation sequencing technology, that is called NGS, and which is concerned about sequencing the mRNA and to get information about genes that are present. And of course, this sequencing technology has led to more clarity in kind of molecular work of the last 1 or 2 decades, so to say. I think previously RNA-seq was not there, and it was done through a technique called microarray. So what essentially is studied for the mRNA-seq, that we are concerned with mRNA-seq. We are not concerned with the other types of RNA. So there is ribosomal RNA, as you all know. There is the more difficult one, that is the tRNA. And although separately speaking, I mean, sequencing is possible for these other types, but particularly for tRNA, it is more complicated because it is very short in terms of the nucleotides, and it has many nucleotide modifications. So some of you, if you remember some fundamental biology, In case of tRNA, you have pseudouracil and so on, I think, as modifications, which make it more complicated to sequence it. However, there are techniques which can do sequencing of the tRNA as well. So, as you all know, that eukaryotic mRNA has a poly A tail at the end. And here is the point where the sequencing technologies have decided that, okay, we will kind of use this factor to proceed with the sequencing further. So that really means that, so in sequencing, the RNA-seq that we do these days, usually it is, let me turn my laser on. So it is done through the Illumina sequencer. And Illumina sequencing, it's very suited for short reads until 300 base pairs. And it goes through a fluorescent coupling cycle. So it shatters the fragments, then it generates reads, and then it proceeds with the further amplification, so on and so forth. Point to be noted here that always cDNA is synthesized from your RNA before it moves to the sequencing steps. So it is not one step. So many people can think that if it is RNA-seq, then directly the RNA is sequenced. It's not like that. So in the sequencing methodology, so to say, oligo DT primers are used. And as the name suggests, so it's a DT, so it's oligos of T's, and it binds to the this poly A tail, and therefore we have the further steps which are involved. There are other ways to do sequencing as well. Something which is very, very reliable on a genome level, whole genome level, that is called Oxford Nanopore. It's very mobile and you can carry it on. And I think in our building where I do research, there is a group who has this device, and it is very easy to do from whole DNA samples. And of course, you have a very specific biosciences, the PacBio, so to say, the sequencing platform, which, as it is shown here, it circularizes the DNA as a pin structure. And then it carries on further with the sequencing. And I think Pacific Biosciences, it's the sequencing platform is more habituated with, or let's say it's more used in case of long reads. It's very specialized for long reads. So I will encapsulate the few steps that we, or at least I, carry out during RNA-seq analysis. So point to be noted here, I will describe all these steps from a plant perspective, because I handle mostly plant samples. And of course, with other samples such as human samples or other organisms, the basic always remains the same. The difference is created how you align that. So the first step, I think, in any kind of this RNA-seq experiment is that you get something from the company which is helping you with the sequencing, something called raw reads. So, okay, so it's given as a FASTQ file, fast and Q. So not a FASTA file, but FASTQ file. So the Q here stands for quality. Okay, and with that we then proceed further with the analysis. So firstly, when you get the raw reads, we have to check that the adapter sequences that are clinged at the end for the poly A tail, that are cleaved or not. So if the adapter sequences are at the end of the poly A tail, they allow the sequencing process, but we have to check whether the adapter sequences has been removed or not. If not, then we remove it through a tool called CutAdapt. I will come to it very shortly. And then, of course, we do a quality check of your data that is there using a tool called FastQC, and then only we can proceed to alignment. And alignment, as you all know, is two kinds. Firstly, for those organisms where a reference transcriptome is already available on a public database, okay? And I think this is the most case with at least stress biologists or plant stress biologists, where we don't work on the evolution or the let's say de novo synthesis of plant genomes, which is another way to do alignment, where we do de novo assembly of the reads, which is very typical, and it leads to sequence where a reference transcriptome is not yet available. And then after alignment, all the steps after alignment is usually done in RStudio. So after alignment, we get something called a count matrix. So until alignment, you still have the sequences, so A, T, T, G, 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 so on and so forth. But after you generate a count matrix, you will have a gene ID, and you will have how many times that gene is there in your target sample. Something we called it as gene count. And with that data, that let's say you have two conditions, let's say stress and control. And in stress sample, you have a count, and in control sample, you have a count. With that information, we proceed further to do differential expression analysis. And upon a successful differential expression analysis, we do data visualization. I will come to that in a moment. That how what is the situation of expression, and how can we expect to have it in front of our own eyes. And of course, at the end, we do a lot of advanced data analysis techniques. I mean, obviously, for the sake of time, I cannot explain everything, but I will try to do a gist or a summary of how it looks like. And I will try to touch upon the important factors here, like gene ontology and a little bit introduction on k-means clustering, etc. So in RNA-seq, there are some important terminologies that people use. So the first is something called Phred score. So it's the quality score of a sequence determination process. And usually we expect Phred score of more than 30 to say that a sample is of high quality in terms of the reads. So if it is 30, that means that one base out of 1,000 bases would be incorrectly inserted. So that's the Phred score. So I haven't seen a Phred score of 40 yet, but Phred score in my case, with I handle plant samples, it is usually 33, 34, 35, in that range, I think 30, in that range. There is a term called RIN value, or RNA integrity value. So as you all know, that eukaryotic RNA, when they are sequenced, so eukaryotic RNA has two bands, So the first is the upper 28S ribosomal RNA band, and second is the lower 18S ribosomal RNA band. And the separation between them, this distance, this is sometimes, this is always analyzed in a machine called bioanalyzer to check your integrity. So to check whether your RNA is degraded or not degraded. So it's a kind of a intrinsic way to determine whether we would proceed with the sequencing itself or not. Okay. And then, of course, we get something known as fragment. That is during the process of sequencing, and you get smaller pieces of oligonucleotides, typically several hundred base pairs in length, and which is being sequenced. And then we come to the concept of reads, as I said at the beginning of my presentation, So reads can be single end. And it is two kinds. The first kind is unpaired. That means the sequence is reported or it's studied from the one end of the fragment, of the nucleotide fragment. Whereas paired read means sequence is studied from the both ends of a fragment. So from a statistical point of view, I think both yield results. It's not that you have to do paired all the time. I think the methodology that is followed for the paired reads that makes it a little bit expensive. So usually paired is a little bit less popular, I must say, as compared to unpaired. And then we come to an important term here. You will always see in RNA-seq papers, RNA-seq methodologies, and in websites, they use a term called logarithm of the fold change. Now, what does it exactly mean in terms of a RNA sequencing study? So we take an example. No? So we take two genes, WRKY and JOX. So let us say that both of these genes, they have an expression under stress and they have an expression under control. And then the fold change is always created by dividing, by making the ratio between stress to control. And then you do a logarithm, and the base is usually 2 of this logarithm. And based on this, you get positive or you get negative. So the first case, the WRKY, you can see easily that it is a positive value. It's plus 2.8. So we can say that in that case, this gene is upregulated in stress with respect to the control. Whereas on the other hand, as you can see in the gene JOX3, it's jasmonate related, it's hormone related. So under stress, the expression reduced as compared to the control. So when we did logarithm of that, we ended up in a negative value. And that's why we can call this gene, that this gene JOX3 is downregulated in stress with respect to the control. This is very important. I mean, we will see when we do the differential expression analysis at some point, that never confuse between control and stress. Is always stress against control, or mutant against wild type, or overexpression against wild type. So it's always, so the reference or the control is always in the denominator. Okay, so if you don't do that, we get very false results because of that. So there are certain prerequisites, and obviously after 2022, after the advent of ChatGPT, many things have been made easy or simple, so to say. And usually, if you want to do everything from scratch, so the data processing, checking for adapters, quality check, and alignment, that is usually done in a shell environment, and then that computer should be really fast and in terms of memory, because that is one of the reasons I could not share the alignment files to you guys, because I think RNA-seq requires a lot of space, and then only you can proceed. Otherwise, if your device is not properly partitioned in terms of space, then you can have problems. And we also need a good quality hard disk storage. And we need a good internet speed, reliable connection to the remote server. So in this case, we'll be using US servers mostly that are usually hosted by Johns Hopkins University. But that also sometimes becomes kind of a factor at what speed or at what time you will get your alignments done. So usually in the past, what I used to do is, if I had really organisms who had huge transcriptomes. So I usually open my computer, assign the task, go home, and next morning I used to take it and process the data. That's how I used to do it, and I always prefer the further steps that includes count matrix generation, differential expression analysis to be done on an Windows computer itself. Although technically speaking, Linux can also be partitioned to be done to do RStudio, but RStudio runs on Linux very, very differently. And one package or one function which runs really nicely in Windows might not run in Linux because of certain issues are there. But obviously, if you are habituated with Linux, you can also run RStudio in Linux. That's not a problem. It's only because of my let's say comfort zone. I have changed that. I always use two computers while working on RNA-seq data, and I keep the Windows part, keep the RStudio part on a Windows computer. Now, people might be wondering that who don't have Linux on their systems, can't it be done? The answer is yes, it can be done. So for that, you can look it up in the internet. There is something called Jupyter, and that's a remote server from where you can connect to Linux very easily from a Windows setup. Yeah. So, and that would make life very easy. I myself am not well versed with Jupyter, but I will show today's tasks or today's processes in Linux computer. So I will switch my, so I will leave this meeting and then switch from another system and then come back actually, essentially. So yeah, so that would be some of the requirements that are needed. And yeah, so then let's get started already, and we can do, I have a test file, test first queue file, and we can get started with that. And then of course, after we do alignment and we see alignment, you guys can ask me questions, and then with a short break, we can start with the next part of our lesson. That should be RStudio. Okay.
Sabarna,
So
sorry to
yeah,
interrupt you in between.
yeah.
So
Mhm.
you are using FastQ file and doing alignment.
Yes.
So is it possible for you to share it with me and the participants can do side by side?
Ma'am, actually, the FastQ files and the alignment I will do in Linux shell, and that
All
might
right.
require a lot of time.
No problem.
But what I have already planned for the participants is a much later step in the differential expression
No
analysis. I will
problem.
let
No
you,
problem.
yeah, yeah. So, ma'am, I would like to now leave here and to join again, if that is okay.
No problem.
Yeah, okay. So I will, yeah, I will leave here. So see you guys in a few seconds. Leave here. I am back in this setup. That's my Linux setup. So I will now share the screen. Can you see my screen?
Yes.
Okay, good. Okay. So welcome to the Linux part of my presentation, so to say. And as you can see, this screen here in front of you, that's the C shell that is sometimes called Unix. And Unix is called C shell because it was developed in the language C. And unfortunately, the system I am using for the last 6 years is in German, but I will try to translate wherever it is necessary. So I will open a new window for you guys to see. And as you can see that it has the computer name while it is written. And from here we start processing the various data or the alignment, so to say. Okay. So the first step that I do for Linux is always to carry on a quick update. So that's the command line that is used, the sudo apt update. Click on yes, and it asks for a password. That's the same password that you use to log into your system. And you click on that, the computer is updating itself in terms of all the algorithms that were there in that environment. And in addition, I also do sudo apt upgrade. Sorry. So that really, I say yes, and that also upgrades all this stuff. I think this will take a little few seconds. I think this step is sometimes crucial because although you might update your computer at regular intervals with the shell scripts, but I guess it is always better to update it before you use it. So now we are ready. Now the important part in the alignment, or before we move to alignment, is that how to let the shell know where your files are. So for that, we use a command called cd, that is called current directory. And I have a folder that is named as zhdc, and that child folder is present in the parent folder called as documents. Okay. So I will first use cd to go to document. Here I am in document, and then I will use cd again to run zhdc. Okay, and then I am in zhdc. Yeah. So here I can really see or really start using the required files, required algorithms to align. Yeah. So for alignment, important stuff is there are many aligners in the available commercially, but I use mostly HiSat2 and TopHat2. These are the two that I use, and for the time being, I also plan to use HiSat2 to show you an example how it really functions. But before that, as I told before, we will do first QC. That is the quality check. The moment I say first QC, I press enter, and this appears. So I hope you can see it. And then I open where my file is. So I go to Documenter, and there should be a place called ZHDC, and there is a file called test run. I click on that and I click OK, and it runs the analysis. I hope everybody can follow me.
They have
Yeah.
seen fast QC, and
Mhm.
I think, yes, they have fast QC in their computers as well.
Yeah.
So one of our I just forgot the name, uh, resource person, Dr.
Mhm.
Sarika Jaiswal, she
Yeah.
made them aware of fast QC
Okay. Mhm.
for
Okay.
editing the reads. I think,
Yeah.
nahi, quality
Yes.
check of reads.
Yes. 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 I think I am also using for the same purpose. So just to show you, I mean, the overview of how the statistics should look like, at the end. Yeah.
All right.
Yeah. Okay. So you can see it is here. The statistics is here. So already you can see the base statistics here, that sequences flagged as poor quality is 0. Yeah. So actually we are good to go, but we can see certain more um, parameters. So here you can see, as I told you before, the Fred score. Yeah, that's the average quality per read, and it's somewhere in between, I think 34, 36, or let's say 34 to 37, somewhere in this range. I think it's the distribution is shown like that, and that means I think we have a let's say go ahead that we can process this data further, and we can go ahead and process this data. And you can see something called sequence duplication levels, and it is being shown with a cross. I think sequence duplication level can mean that similar regions of genes at certain points of time, but I don't really consider sequence duplication to be a really flaw in your data quality when it comes to FastQ. And we also have something called overrepresented sequence. We can hold a discussion at the end when we talk about this, when you guys can ask me questions, and we can discuss further about overrepresented sequences. Interestingly, the adapter here has to be removed. Yeah, because there is a little here. You can see this red. This is called Illumina Universal Adapter, which is there in red, and it has to be removed. I will show you the command which is used to cut the adapter and then proceed further. Unfortunately, these all things I cannot show you everything in detail in the Linux script, because sometimes the running times might vary. So yeah, okay. So that would be it with the quality check. I think I'm satisfied with the quality, and then we can start with the alignment itself. Yes. So we write the name of the aligner, that is HISAT2, and we click here, and then the computer says you that how to use it. Yeah. So see the top here that we did not give any inputs yet, so that's why it is kind of an error message here. And it also tells you that HISAT2 version by Devon Kim, that is the last known operating base of where HISAT2 was developed. And after that it says you how to use HISAT2. Yeah. And in terms of the command itself, so first you are going to use HISAT2, then you are going to put a -x. This stuff that you are seeing here, this is called an index file, to everyone that is present here. So the computer cannot read an annotation file, okay, directly. It always uses an index. Now, the index file that it is prepared, it's from the chromosomes, okay. So usually for plants, we have plant ensemble website, and from that we download the individual chromosome files, and then we concatenate it using the cat command. It's a very small command, and we concatenate it, and then we run HISAT2 in the build mode. Okay, so it will be HISAT2 - build, and then you put your concatenated all the chromosome files, and then you get an index. Fortunately enough, this can take up to 4 hours. So I did it yesterday, this index generation, and this is already there in this folder that I am currently in. This is ZHDC. Actually, this processing time all depends on the server, how it is processing, but it can take really huge amount of times. And same for the alignment itself. Okay, so we move ahead. So here you see -1 and you see M1. Here you see -2 and you see M2. So this entire thing is made for a paired and read. That means from both the directions. So mate1 is from forward direction sequence, and this mate2 is from reverse direction sequence. Or alternatively, you can use U caps U, and then you can use your unpaired read, which I will use because I have the test file which is unpaired, and then you have to write to the computer that what is going to be the output file name of your stuff, and usually then only you can get the output. So here you can say anything, so test output.sam. Yeah, so I will shortly go back to the folder directory because I will need to check the exact name. Can you see my screen?
Yes.
Okay, in terms of the, yeah, so I am now in the Explorer, I'm back in the Explorer. So to all, these are the index files. Yeah, so index files are in total 8. Okay, and sometimes it is difficult to predict because usually the fast queue that I am going to process, that is from Arabidopsis, and Arabidopsis has 5 chromosomes. So it should not be 8, but these are some more information that is required for the aligner itself, that is required for HISAT2 itself, and that has nothing to do per se with your chromosome or with your plant. Yeah, so that has nothing to do, but I just opened it to look at the name. So it is ht2_index_fin. So just in case I forget, I have also to write it down. And the name of the file is testrun4.fastq. So with that said, let's go ahead with the alignment. So I am here. So I will write HISAT2 x, name of the index is ht2 index underscore fin. Sorry, my caps is on. And then we write U test 4.fastq, and we write S test output.sam. So let me give you a quick check whether I have written everything correctly or not. Yeah, I have written everything correctly, so hopefully it should work. Then I press enter. Now it has started. So we wait a few minutes before it gives you the output, and then we can proceed further. I think that's one of the probable disasters that data analysis has, and particularly with C, need a lot of running time sometimes. So I hope that in 5-10 minutes it should be over. I guess we will wait for 5 more minutes because it should be done, I guess. Okay, I believe since it is taking a lot of time, I can show you in parallel how to get the let's say the reference transcriptome. At least for plants, I can show you that. So I'll go back here. So you can see here. So I will go here just a second, and I will type here ensemble plants FTP. Click here, then you go here, and you can see here that this is the site where you get all the required data, and then you click here as the FASTA, and you get all the chromosomes. And people who are seeing it might also ponder upon why you get a .gz. So .gz essentially means that it is compressed, and you need to decompress before you start using it. So essentially, you get all these as individual FASTA files, and you have also here a separate mitochondrial file and a separate chloroplast file, and then you just combine them together. And also, at the beginning, I talked a little about the annotation file. So you can see here that what is written here, that it says gene sets, and you can download either one of them, either it's GTF or it's GFF3. So this is required at later on step to make the algorithm understand which part of a sequence or which portion of a sequence belong to which gene. Otherwise, at the end, you will get gene 1 count this, gene 2 count this, and so on. So this is also another file, a big file, and takes some time to download, and then also you can download these as well. I will go back already to my, so you can see my screen here, and what you can see here is that the alignment is over. So let's go through the results a little bit. So what does it say here? That you have total this amount of reads, and of this, how much was aligning one time and how much was aligning multiple times. That means, let's say same gene, but different regions are getting different kind of alignment. So overall, that's the 70.54% alignment rate. And point to be noted here is that unless and until the alignment rate reaches or exceeds, let's say 70%, we cannot go further to do a nice quality differential expression analysis. So that's really important when we do this kind of analysis. And I think with this, I already did the necessary BAM files, the necessary alignment files that are required for the next step. So you guys must be wondering that the output of the alignment is SAM. It's called simple alignment map, and if you want to go further, it can also work. But SAM files, the problem with SAM files is that they take up a lot of space in your disk, and you might, with increasing amount of samples, you might risk kind of freezing your system. So it is always better to use something called SAM tools here, and then convert a SAM to a BAM, and vice versa also can be SAM to a BAM is. And so I write SAM tools, and you can see how you should use it. And yeah, so I already did the necessary BAM files, the necessary SAM files conversion yesterday evening, it took a lot of time, and I already made it ready for you guys to, so that because after this we are going to use this for gene count generation, and that can also take a lot of time. Unfortunately, my apologies that BAM files are a little bit difficult to be shared even by cloud, because this is total of I think 8 GB of data, and I was facing some issues yesterday. So what I will do now is, now I will stop here maybe at this point to listen to your questions that you can have with all that I have spoken so far. And after that we can have a break, and after the break I think we can uh, come back again to join to the art session. For the art session, I will do one step. I will share the data around with Ruchi ma'am, and ma'am can send around to everyone, and then we can do an actual hands-on with the data set. So with that, I think now I will be open to your questions. Um, you can ask me questions on what we have done so far.
Sabarna, I must
Yes.
say,
Mm-hmm.
you are such a good teacher. Like you
Okay.
have explained everything with the very nice pace. It was very nice. Everything was understandable.
Okay. Yeah. So thank you very much, ma'am. I am really keen for any questions that people might have.
Actually, maybe in hands-on session they will have some question. So for
Mm-hmm.
now, we can take 15 minutes break. Is it fine,
Yes.
Sabarna?
Yes, we can
All
take
right.
15 or let's say 20 minutes break. So
Or
now
okay,
the
20 minutes break.
so now it is seven. Now it is 6:35. Am I correct? No,
It's
5:35.
5:35 here.
Yeah. So 5:35. So let's say five
Yes,
55. We sit together.
no problem. Correct.
Yeah.
And in the
Yeah.
meantime, you can share file with me. I will email to everyone.
No, ma'am. I will. So that is one more step in our. I will show it. Then I will share it
Mhm.
to you.
No
That's
problem.
an.
No
I think
problem.
that's an TXT file. I will share. Yes. Mhm.
All right.
Because yesterday I spent some time in 8 gigabytes of data, and that was really difficult. So that's why I decided that I will have this all ready, and then we do the further processes. Yeah. So, ma'am, I think we see each other in 20 minutes.
All right.
Yeah. Okay. Okay.
Oh.
Yes.
Sabarna, I think we can start.
Yes. Yes. I'm just
All
waiting for
right.
my art studio to open. That's all. It will take 2 more minutes for my art studio to open, so I will turn my mic on when it opens. I am ready, I think. Um, yeah.
Yes,
So
we can start.
yes. So welcome everyone to the next part of the talk, which we will run in Art Studio. So let's get started with it. I will share my screen. Yes, see my art screen. I think is visible.
Yes, it's visible. Just click on hide so that we can see
Yes.
everything.
Mhm.
Yes.
Yes. Okay. Okay. So, I think many of you have already installed RStudio before, or have RStudio already installed. So, just a quick introduction about R. R is another programming language, and as you can see, the console, the console where you can see its screen is a slightly different from what you saw just earlier. It was C. It's much old language, so that's why less user friendly. And this is RStudio, newer language, more user friendly, and one of my favorites, to be very honest. I think I will always put R ahead of Python, because Python is more mathematical and statistical. So as you can see, this is the area where we write the codes for R, and you can see a whole lot of options here, like file, edit, code, view, plot, session, and so on and so forth. Now, just to give you a short reminder here, that R also needs to know where you are running the files. So what step are we in right now? So we had finished the alignment, yeah? And after the alignment, as I told you guys before, that he said two outputs the alignment files in a format called SAM. Yeah, and SAM takes up a lot of space. That's why I had already changed that to BAM, that's binary alignment map. So whatever was taking space up in the SAM format now has been changed to binary format. Yeah, and as always, R also needs to understand where you are, where your files are. Yes. So what we are going to do now, we have the BAM files. And if you remember, I also showed you a file called GTF, that is the annotation file. Yes. Now, in your BAM files, the genes are still in A, T, C, 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 G, G, so on. Yeah. It is not in the gene ID versus count format yet. Now, which software does this job? This is called, this package is called rsubread, as you can see here. Yeah, this is called rsubread. And so we will use a function from rsubread, which is called feature counts. And in this case, feature means genes. Yeah, and I will finish the feature counts in my computer. I will generate an output and I will share it to Ruchi ma'am, and she can share it to everyone around, and then we can start the actual differential expression analysis that will be hands-on. Yeah, and for each step of the differential expression analysis, I will share the chunk of code that is needed in the chat room. If it is, I think that will be more easier. Although I had shared a script, the script also should have the code, but just to make it more easier for everyone who is participating, so that we can follow, we can do one-to-one. And if you have questions, you can always let me know. So let's get started with feature counts, and I will call, so R has library system, and each library encodes different kind of packages. Now this feature counts is present in the rsubread package here. Okay, so I have already installed rsubread package. For those of you who want to install it, I think you can go to here in the tools, and you can install packages and write the name as rsubread. It will, a dialog box will come up, yes, here, and you can here click on write rsubread very easily, and then it will install the package along with the dependencies. So dependencies are basically some more associated functions that are connected with this package. Yeah, so I will do this part in my computer, and the later on packages which are required for differential expression analysis, that I will give you guys a little bit time to install the packages as well. Okay, so let's get started. So I will call the function library rsubread. You are seeing here a script of R. Yeah, So if you go to file, you see here, new file, you can see many kind of scripts. So you have R script, you have R notebook, you have R markdown. All are essentially the same with some kind of differences in between, but the easiest to use is the R script for this kind of analysis. Yeah. So if you click on get WD, what does get WD mean here? So WD stands for working directory. Yeah. So if you click here, it says that my working directory is in documents, which is not sufficient, unfortunately, because the BAM files, the alignment files, the annotation files, these are, just because they are huge in size, I have kept it to an external hard disk. So I will change the directory right now. Go to session, go to set working directory, and you go to choose directory here. And then this is my external hard drive. And then I click on the name of the file that is there. And then I click here, and then I click open. Yeah. So now the directory is set to the exact location where the files are. Now let's get back to the command that is used. No? So as you can see here, there is a command here which says BAM files list.files pattern equals to BAM ignore.case equals to true. That means at this point R is wanting you to go to your directory and list all the BAM files that are there in that directory. Okay. The same for the GTF file, as you can see here. Same thing. It wants a list of the GTF files. So we will run it accordingly. Okay, and you will see the output very soon. So if you see here, BAM files, I hope everybody can see me. So BAM files, it says CHR character 1 to 16. So I have 16 files that I have kept for the analysis. These are various BAM files. So we have wild type control, we have wild type drought, we have mutant control, we have mutant drought. So 4 conditions and 4 replicates each. Now those of you might be wondering what are replicates. So in RNA-seq, it's never done on one sample. So if there is one condition, let's say there is stress needs to have at least 3 biological replicates. Same for control needs to have at least 3 biological replicates, and we have one more. This is just a dummy data set just created for this purpose. Yeah, just used for this today's talk. So as you can see, this is already done. So you have BAM files, you have GTF file. Yeah, and then I will already run this command. Okay, so what this command says is it is named as fclim, as you can see here, and then it calls the function feature counts. Yeah, then it also takes into account the BAM files, then the exon. This comes from the annotation file, guys. This just few moments back I loaded the GTF file, that was the gene transfer format. This exon, or which portion is an exon, these all attributes are present in the GTF file. Okay, and we also need a gene ID. That is essential for us, because otherwise we cannot identify which gene is what. Okay, and then also we are saying that we are using a GTF annotation file just designed for our target organism. In our case, the target organism is Arabidopsis. Yeah, so we run this command. Yes, just a second. I think there has been some problem because I might have forgotten to call the rsubread package. I think now it should not be there. So now it's running. Yes, And we will wait for 2-3 minutes before it finishes its run, and then we will write the output, and this output I will share with Ruchi ma'am, and she can share with everyone. I think it will take a little bit more time, maybe 5 minutes, and we will be over. Let me check. Oh, fortunately, I think I might have the output which I ran yesterday. So in that case, I can share it with you. Yeah, I have it.
कल की नहीं है वो? अच्छा, अभी कहाँ पे हो आप? अच्छा, कब तक निकलना है? चलो मैं देखती हूँ, नहीं तो बता ही देती वैसे।
I think everyone, I think I should be done in few minutes. Should be done. In the meantime, I have shared the counts file already with ma'am. And ma'am, it will be nice if you can share it around.
I'm just sharing it with them. Just a
Okay.
moment.
Yeah, yeah.
Sent to everyone.
Okay, okay,
It's yes, sent. Please check your spam also, because last time, the mail went to the spam. Anyone, please confirm that you have got it or not. Sabarna, they have got it.
okay, okay. Good. Then I will get back here. It is still one more minute, maybe. Then I can explain in details.
हेलो हाँ कहाँ पे हो
Just the last five, I think
अच्छा
second
कितनी
last
देर में
five
चाहिए
minutes.
गाड़ी हम्म 20 मिनट का फर्क है भाई दोनों के बीच में अभी आप कहाँ पे हो
3 minutes.
अच्छा तो मेट्रो स्टेशन से कितना टाइम लगता है मैं वो अच्छा ठीक हेलो हाँ अभी मेट्रो पहुंची है वो तो 6:50 के अराउंड कह रही है कि वहाँ पे है तो उसके हिसाब से देख लो अगर आप निकलोगे तो फिर उसके
Resume my mic once it is complete.
चलो
Manisha, please check the spam.
So it's over. So what we had done in the couple of minutes is we had fed the alignment files into feature counts, and we told feature counts to change it to a table. This table should have the gene ID and followed by all the samples that were used. So everyone who are looking at my output right now, it is a TXT output to be very precise. It is a TXT output, and that means it's a notepad file, and it has the gene ID for wild type C. You can see wild type C, 4 files for wild type C, 4 samples for wild type C, 4 samples for wild type D, 4 samples for mute C, that is the mutant, and 4 samples for mute D, that is the treatment. Okay. And then we can further start it. Yeah. So before we go to the HR part, I will do certain stuff with the notepad file. Okay. So please let me know, whoever is doing it with me, please let me know whether you can follow me or not. So you can open the notepad file. So the notepad file is here. This is the notepad file. And as you can see that there is gene ID and there is length. I'm sorry, I forgot to mention that. And so we have to little bit modify this notepad file. So you can simply press Ctrl A, then Ctrl C, and then go to Excel. And paste it. And if you paste it here, you get an Excel file with all these guys. Yes, we have to do certain changes. Okay, everyone present here. So firstly, for the time being, we don't need the length file. Okay, because we are handling directly differential expression data. Point to be noted here, length file could have been used if we wanted to generate transcripts per million, that is TPMs. Okay, that's the gene expression estimates. Right now, what you are seeing for each of the genes that you see as gene ID, it's how many times they are present within your samples. Okay, and to simplify it and to see the time constraint, we will work with the wild type only for the time being. I mean, don't delete it right away, you can keep it, but for the time being we are going to work with the wild type samples. So you can put them away, put it to a new sheet or delete it, something like that. So I will just delete them. And now you have on your screen all the wild type samples. Yeah, all the wild type samples, you have it on your screen. So I'm just zooming in. So then you just remove the .bam extension from each of them. I'm just removing all the BAM extensions. Okay. And then you can go to, sorry, the link has not been removed. Delete. So it's deleted. Then you can go to file. You can do save as. And here you can choose a place. Okay. So I will do browse. And I will choose my directory, which is already set. Okay. As Samsung T5. That's my external hard disk, and then I will click to my folder, and here I will paste it, not as a Excel workbook but as a TXT file. Okay, So let's give it the name ZHDC, let's say 1, something like that, yeah. And then we have to do text, tab delimited, okay, and then you can save it. Okay, that will be the first round of doing this, and I will return to the this thing. Can be gone. Good. So I will return to the R file, and I will also return to my screen. So at this juncture, people who are doing this, please give me. You can raise your hands if you have done until where I have done. Okay, okay, great. So I think we can proceed to the next part here, which is the differential expression analysis, guys. So I will share you chunks of codes, although the codes are already there, but it is always better to do it quickly. So here I am with the first set of codes. So I will copy the entire stuff, at least until where we needed it, and I will explain to you what to do next. So
Sabarna,
everyone, if you can,
please
yes,
pause for a moment, please. Uh,
yes.
Participants, did you not get the file? But they are doing side by side. Go ahead.
Okay. Okay, then I proceed. So, guys, whoever are following me, I have shared a chunk of code. Let's break it step by step so that you don't face problems. So, this first chunk of the code is related to differential expression analysis, and we require certain packages to be installed. That's why I will pause here for 5-10 minutes. So, we need the following packages, yeah, Lima, HR, tidyverse, and ggplot2. Once again, Lima, HR, tidyverse, and ggplot2. You can directly go to tools, go to install packages, and then type in the name, all of them, and do install. Okay, it shouldn't take more than 10 minutes, and only that I will be able to proceed further. Okay, so people who are doing it alongside me, please do these packages as I showed it to you right now, and confirm whether it is done or not. If you have installed already the packages, just raise your hand or give a thumbs up. I can understand from it. And don't forget, please let me know if you have some package installation problems. We can also take a look if you have package installation issues. Sometimes it can happen actually.
No, it's fine downloading.
Okay, good. Should be fine, I think. I mean, I can wait a few more minutes for the others. Should not be a problem.
Sir, can you please repeat once how to download the packages?
Sure. So if you go back to our studio, you can see here tools, right? And you go to install packages. Okay. And then you just write here your package that is enlisted in the chat. So first one is Lima. You can write. So Lima, sorry. And you can write on install dependencies and then you can click on install. Okay. That should
Okay,
work
sir.
actually. Okay, good.
Students just see the spelling from the chat box. Library lemma, edge r, teddyverse, ggplot2. Sabarna, is there is any way that we can make out that the packages are being installed? Like, is it done or not?
Sir, it's showing package HR is not available.
Okay, in that case, I think there is a separate way to install Azure. If your this thing is showing error in that way, just see my screen. And then simply you write. So there is I think they update every time if it is not installed. So you can just write HR installation. Okay, then go to there. So you can see a chunk of code here. See here, this is what I mean. Okay, I can share it with you in the chat box quickly so that you can do that. Okay, and
Yes, sir.
yeah, so just a minute, I will share it. Just copy that and paste it there. So essentially what it is doing is your package installation failed because your local computer could not manage that. Then what you can do is there is a remote manager for R packages, something called as BioC manager. You can connect to it and it will give you access to that package. Yeah, try it this way. And also try with other packages. If Azure is giving this issue, in your case I think you are Bhumi, right? So I think you can also try it for Lima and other packages if it's giving the similar issues. Just ignore the name Azure and then put the package name there. Okay, that should solve the problem.
Yes, sir.
कोई भी बना दो,
Okay.
कोई भी बना दो, उसको किसी डब्बे में डाल के रख दो।
Is everyone done? Can I proceed to the next steps?
Yes, Sabarna, you proceed.
Okay, good. Okay, then I will proceed with the next steps. No, so I will open my. Okay, so I will also call these libraries. So people who have installed the packages, after the installation, you have to call the libraries. Okay, otherwise R might get confused. Okay, so moving on. So my libraries are installed here, and as you can see, there is another command called getwd. That means it wants to understand the working directory once again. Okay, so I have by default chosen the working directory. Just a second, in the my working disk, in the hard disk. So I will do that once again, and I will set it now. So it is set, yeah. So, and then if you run this getwd, that means just to be confirmed that your working directory is correct, what you just set, okay. And then if you see the other part of the code, that it says that counts is once the name of the file. So I think I have put zgc1.txt. So read.table, guys, is essentially that it wants to read your file, the txt file that is there, and it can only read that if you can successfully set it to your current working directory. Okay. Header equals to true means it recognizes column names, and then we can do it like this. Okay, as you can see, in my case, it already appeared like it is, and a variable in the name of counts has been created, which read in the data from the txt file that we just created. Now, if you want, we can skip this command. This is basically just, how is it called, this streamlining of the document itself. It's a very internal factor. This is not necessary. I had used it before, but if you guys want, you can skip it. Okay. So that was the step, and then we move on to the next part. Just a minute. I have to see until where I have shared you the commands. So I have shared until library size. So let's do the library size here. So library size essentially is the CDNA library depth that was created for all of your files. Yeah. This is taken into consideration because this is a technical factor that helps the algorithm decide what should be the expression values. Okay. And I think until here I had shared you the commands. So until here, if you manage doing it, so library size variable should be created. Okay. And then I will tell you the next steps, which is the plotting of the MDS. I can share it to you on chat as well, people who are following me. This is the MDS plot, and if you run this command, then on the right side, guys, you can see that it shows you the separation between the various samples. And as I said to you, that in RNA-seq, the important factor sometimes is knowing the quality of your samples, the homogeneity of your samples. So what essentially this plot is doing. So MDS stands for multi-dimensional scaling plot. It will take all your samples and do some dimensions. And based on that dimensions, it does some statistical calculations, and it does a kind of a plot, as you see here. And you see that most of the D samples, except one, is on the right. Yeah. And most of the C samples is on the left. Most of them. Okay. There are outliers, it looks like, but most of them are clearly separated. Okay, there is not much of a overlap between the various treatments. This is very important to carry the analysis further, because this shows that your control samples have a variance, a general variance, and your drought samples, which is the stress samples, have a different kind of variance. Okay, so it does not overlap, and this sometimes can also mean that you are going to get good quality of differentially expressed genes. This is one of the reasons why MDS plot, a multi-dimensional scaling plot, is very essential before you proceed to the next steps of the differential expression analysis. Okay, so I can wait here 1 minute and you guys can share what you have done. People who have generated the MDS plots, you can raise your hand if you want, or give a thumbs up.
Sabarna, I think this downloading packages may be a problem for the participants. So, because we are recording this, so you can just explain and they can repeat whatever you have taught. Now they have files. Once they will download the packages and understand the stuff.
Okay, good. Okay, then I can continue. Yes.
Yes.
Okay. Okay, so now that we have done the MDS plots, let's proceed to the next parts of the analysis. So, which is very important. So, we have to now tell the computer that how many are controls and how many are stressed, or let's say treated. Yeah. So, this will be changed to, sorry, so this will remain as control and this will be 4 because we have 4 repeats of that, and drought will have 4 repeats. Okay, this is also important. And then you can see control enter, and it has a TRT files. Okay, that means it has 2 levels of conditions. One is control and the other one is drought. Okay, and then moving on to the next step, we need to create a matrix based on what we defined as control and what we defined as drought. So it creates a matrix. Okay, and next we go to a very crucial step that is called VOOM. People who have studied RNA-seq before or who have seen papers concerning RNA-seq. So VOOM and TMM, these are 2 very technical terms that are used for data normalization. Unfortunately, I cannot share the mathematics behind it right now because otherwise you guys will be very overwhelmed. But it's just mathematical, it's just a way of normalizing the data. And it uses something called CPM, it's called counts per million, and it changes it to another log variation that is called log base 2. Okay, and it's a little bit tedious to understand it, unfortunately, but I will skip the very meticulous mathematical details and just proceed with the command itself. So as you can see, we call a variable that is called V, and we use this symbol. As you can see, we have used it before as well. And we call the function called VOOM. It's called a normalization function. We feed in the counts file, we feed in the library size, and we fill in the factors that were there. So in this case, we have 2 factors. One factor is control and one factor is drought. Okay, is it clear? So it will generate a linear plot, should be linear. Yeah, so as you can see, the plot on the right hand side, see, it starts as a curve, but it goes down as a kind of a straight line. Yeah, it's not a fully straight line, but it's kind of a straight line. I mean, it's kind of a line, so to say. It's not perfect. You can see some scatter plots which dots which are just deviating. It's not perfect, but it should be linearly, because VOOM at the end of the day is a linear normalization function. Okay, it has a tendency of producing a kind of a straight line for the normalization. Yeah. And then we go to another mode of normalization that is called TMM. Okay, and I just will call these functions. These functions, by the way, guys, are there in the script that has been sent already. and I will just call this function. So what you can see here is, guys, if you see here carefully, so you can see control, there are 4 ones, and in droughts, there are 4 ones. So that means the the algorithm has already allocated 4 kinds of repetitions for each of control and each of drought. Yeah. And with this information, we can proceed further. Stuff here. So we will do just a second. So we will do something called Bayesian fit. It is also important that it completes the normalization. Unfortunately, with these meticulous details, you guys might be a little bit overwhelmed, but I cannot help it because these are very crucial to the scenario that we have to do differential expression analysis, and without normalizing the data using advanced statistics, sometimes what you get at the end could not be trusted and it becomes false positives. So for that, certain very strict and rigorous steps are always needed. Yeah. So I will do this here. So I will click here because we named it as drought. These all code is there in your script.
I would request my participant that they should copy these codes, but because once the meeting is over, the codes will be gone. Right? So please copy it somewhere. Sabarna, will it do copying the code somewhere and then use later?
Yes, it should work. And the script that I gave you, ma'am, also has the code. So it should not be a problem.
All right.
Yeah. So, yeah, so this is the contrast matrix. Here we are setting contrast. Here also a point to be noted here that very important and very, very important. We cannot do the mistake here, control versus drought. If you do so, your entire analysis gets jeopardized. Okay, so be very, very careful at this point. It's always stress versus control. So this is generated as a contrast matrix. So this we also have to do as a final step. So now we are here at a variable called results. And these results really tell you at the end how many are upregulated, how many are downregulated, and how many are non-significant. Okay, I hope I am clear with this because it is also essential. Now, this decide tests function, it, as you can see here, I will maybe scroll it through. So it has something called FDR. Okay, that's called false discovery rate. So FDR, in your mind, you can set it that it is a improved version of a p-value. If p-value is 0.05, less than 0.05, we take something as significant. If it is greater, we take something as non-significant, right? And FDR is a better version of p-value in the sense that p-value has some false positive, so it's not always perfect. In order to do that, these guys, Benjamini and Hochberg, these were the original scientists, that they discovered a way to clear false positives from the raw p-value. Okay, and here we can also see that there is a parameter called LFC. Okay, as I explained to you in the start of my presentation, that LFC means log-fold change. Now, if you want, we can filter our results that we are going to see in a moment based on log FC and p-value. But since I think we are all beginners at this point, so I will remove this parameter, and we will see how much fits to our, how much we are getting as significantly expressed genes. So once again, revising this stuff. So if FDR is less than 0.05, that means a gene has changed significantly under stress condition as compared to the control condition. Now, change significantly can mean it can go in an upwards direction, it can be up regulated, or it can be down regulated. And if a gene has FDR more than 0.05, which means that it is unchanged. So it did not have a change in the stress condition. Okay. So with that, we can run this command and then you can see some of the results. So it is very less, guys, because I only chose a very dummy data just to show you how the thing really looks like. So it's very less, but you can see that there are 53 down regulated and there are 10 upregulated. One of the reasons why I wanted to choose this data set is that this data also shows you why there might be a reason that you are getting so less amount of up or down regulated genes. One of the reason can be, if you remember, when we generated the MDS plots, remember I told you that there was one replicate from the drought which was sitting in the control, in the wild type control. Yeah, this can be one of the possible reasons that it has this value. So what we do is, since we have 4 biological replicates, we omit one which was creating this problem and we go with the 3. So minimum you need 3 and then we do the analysis further. Yeah, and just I wanted to show how it looks like and how things can be done out of it. So let's get the result in all. So I will create this. So wait a second, just a moment. So I will just rename zhdc test.txt, yeah, and then I will call this function and it will write the table where I have chosen my working directory. Yeah, so it should be something like this. Okay, then if I go to the my working directory, see, you can see already ZHDC test. That means all your result of the differential expression analysis that were to be generated are now generated. Okay, I will open it. Okay, then I have to copy it. I have to do Control C and then I go to an Excel sheet, an Excel file here. I will open my Excel file and I will try to paste it. Sometimes it doesn't. Yeah, it is not happening, which means I have to use my Linux computer because the TXT files sometimes are in this format, because you can see, guys, that there is a double this opening, this quotation here, which hinders Windows processing stuff. So I will quickly transfer this file to my Linux computer and quickly make a conversion so that you guys can see what can be done further. So I will take this file with me, my disk with me. Just bear with me for a second.
अस्सलाम वालेकुम। आ गई जैनब? आप दूसरे से आए हो? मैं चिली चिकन बना रही हूँ। चिली चिकन, वैसे दाल चावल भी है, सब्जी भी है, चिली चिकन भी है। अभी ब्रेड ऑमलेट खाना है? हाँ। दे दो।
So I am done with the file conversion. So I will show it to you on my screen how the file now looks like. So if you guys can see my screen, so you can see that now everything has fitted within A, B, C, D, and E, which was creating problems for the text file. Another point to be noted, I think, once you guys, whoever will take up RNA-seq analysis at some point in their lives, I think what it will teach you to how to handle different file types in Windows, and there are certain weird processes which you will learn eventually how to deal with them, because not only then you will learn programming, I think then you will also become an expert in the Windows file types. Yeah. So I have the gene ID, I have the log FC, I have raw p value, which we don't need right now because we will do everything based on the FDR. As you can see, that the number of significant genes, chain genes are less, and it is filtered through the help of FDR. So I will also take the help of FDR, and I will take where it leads me. So I will stop here because this is less than 0.05. I am not taking less than equals to. You can also do it, but I am taking less than 0.05. Okay, that's why I stopped before this 0.05. Okay, so I do Control C and I paste it here. Okay, so this will be the first results of what we obtained as RNA-seq data analysis, and we have significantly expressed down-regulated genes and also some, I think, up-regulated genes as well with the data, dummy data that I provided with you guys. And you can see that also the top one is the most down-regulated one with the most less FDR. So e raised to the power minus 5, as you guys know, e is the exponential, that's e. If you guys have ever used the scientific calculator, you can also see e as this caps e, the small letter e. So that would be it, and this was one of the first results that we got with this data. Now, one further step that I can still show, because we still have some time, and which was not included in my script, but this is very easy because this can be done online. Okay, for this you don't need to do any kind of coding for this step that I'm going to show. So imagine a situation where the number of significantly different genes were more than 1,000, then obviously it is not feasible and it is not possible to identify each gene painfully by hand. I think I am clear here. So if you have more differentially expressed genes, now you also want to maybe know that, hey, are there pathways that these genes might be involved in? And if so, how should we know that? So there is a process that is called gene ontology analysis. Okay, so what you need to do is you can copy these significant genes. I mean, ideally, it would be better to separate it on minus and plus. You do gene ontology with plus log FC separately. That means you are doing with upregulation separately, and you are doing with the minus regulation separately. So you are doing with down regulation separately. But for the time being, I have chosen all of this together. I do a Control-C quickly, and then I go to Google, and then I go to here, and I write Shiny Go. That's the one of the easiest web servers that can easily help you to do gene ontology analysis. There are other web servers that's called, I think for humans there is one called Gorilla, and then there is one called gprofiler, and many you can see that on the web. But for plants and for me, I think I am more habituated with ShinyGo. So you can click here. Sometimes the first version doesn't work, so you have to go with the down version 0.77. So you just click here, paste your gene, then go to change species, and we are handling, I guess, Arabidopsis, so we will change it accordingly. Yeah, so here I can write Arabidopsis. Yeah, you can choose the first one. And you can go to submit. Okay, let's see what happens. Okay, so if you see click on biological process, there are no enrichment. Usually, sometimes it shows some patterns here. So what essentially I did right now, so I asked the server to give me top pathways. Okay, now here, what does top pathways really mean? So you have submitted a list of gene IDs. Now the web server calculates something called as fold enrichment. Okay, that means in the Arabidopsis reference transcriptome, there are already annotated genes belonging to one pathway. So let's say the first one, the N-dimethyl aniline monooxygenase activity, that pathway already has some genes in the reference transcriptomes. Now, comparing your list that you provide and comparing with the reference transcriptome list, it generates a variable called fold enrichment. Okay, and with this, the top is of course this N-dimethyl aniline monooxygenase activity. You can also visualize this with the help of a chart. The chart is something like this. You can play it around. You can, so since this rectangle is very small, you can just play it around and increase the aspect ratio. It becomes like this. And if you don't want this gray background, go here and you click on classic, something like that. It becomes like this. And you can also play around with the circle size, something like this. Yeah, something like this, let's say. So in all, I think that's what gene ontology is. And yeah, so that's what it really shows. I think there is a legend of the figure at the end. So the dots that you are seeing here, the size of the dots, that means the number of genes present, and minus log 10 FDR, the higher the value, the more significant it is. Okay, That's how gene ontology is usually done. I mean, we can still play around. I mean, there are chart types, and you can do whatever you want to do with it. And I think there are bar plots as well, and so on. I think so. You can play it around, and there are multiple, ample other options and ample other ways how you can make this gene ontology. And I think this part was not there in your script, so I took the opportunity to show it to you how it can be done. And last but not the least, let's try to figure out how to do a volcano plot. For that, so volcano plot, as the name suggests, it's shaped like an inverted volcano. It's not a complete volcano, so it will be shaped like an inverted volcano, and it will show with colors how many genes are upregulated, how many genes are downregulated, and how many genes are non-significant. Okay, that are not changed with respect to the control. So with that, I will take this. I have to delete that. Yeah. So I will keep this, and then I will save it as series DC. And I will go back to our studio. I will go back. I think this command has been already been shared in the script that I had shared with you guys. Basically, I think you guys might have the question that how am I deciding which commands work, and how do I decide if a code goes wrong, at what point it should be changed, at what point it can be streamlined, and so on. So this, I think this doesn't have a direct answer to it. It's always, I think, trial and error. And of course, there are some online websites which can help you to get an optimum code which works all the time. Okay, so I will go to the code because I keep that handy time to time.
Deep breath.
So here is the piece of code for the volcano plot, and volcano plot can be done in ample number of ways. This is one way which I prefer to do it. So first, what I need to do is I need to import the dataset. Yeah. So I import the dataset from Excel that is there in my working folder that was there, and if I go here directly, I think yeah. So it should be here. I have to be a little patient because it takes a little bit more time. So it is reading in the data that is necessary. So I import it.
मत खिलाना, इसको एलर्जी है.
Okay, so it says that it cannot take in. Let me solve the issue. Yeah. Now it took the file, so I can proceed. So essentially it also uses ggplot and ggrepel. I will change the file name because it has to be changed. Then I am putting, so here interesting to see that there is a dollar symbol. So the dollar symbol means you are inserting something in the Excel sheet. And firstly, I name it as NS, that means non-significant. I run this code, and then I have to change certain stuff here because here we are defining what is, let's say, what is differentially expressed or not. So this definition has to be really be perfect. So this means click on DC and we click on log. I think it was just log FC, I believe. Log FC. And
Naira,
also with the FDR,
Naira,
same thing. So let me change
1941.
it
19 is
to
7,
that.
na? 19.
And FDR
1941.
0.05, and that means
741.
upregulation. So essentially, we are
Naira,
saying the command,
Naira.
deciding the necessary criteria for this. So, and if it is less than -1, it is down regulated, and so on. So I will do. So it should be here. So let me cross confirm whether it's correct or not. Yes, it's correct. So then I run this. Hopefully it should work. Okay.
खोल दो जरा.
Yeah, took it. Now we are going to plot that. So with standard ggplot, we're going to plot it, and data is zhtc. Should be log fc. Sorry, is the x-axis. Y is the FDR, and yeah. So this has slightly range of colors. We'll see how it fits to it. Okay, so let's see what is the outcome. Just a second, I pressed the wrong button. Yeah, so there's some issue with the code. So, So, guys, sorry, it's I think my mistake. I only uploaded the various significantly changed genes. That's why the non-significant was not coming. But just allow me a few more minutes if I can. Yeah, but I think it can be still. This code is still functional. So what happened was, I only took the significantly expressed genes, and that's why it is not showing. But if I change the colors, maybe something will strike. So let's call the blue as down regulated, and let's call the red as up regulated. So still you can see the changes. Yeah, something in that direction. So um, and why the shape is still not complete is that I forgot to put in the non-significant genes, but I think we have still time left. We can still try to do that, just to show you how at the end it should look like, and otherwise it doesn't make sense. Okay, just give me 2 minutes. I think. I think the problem could be solved. And I have the correct file right now. So I can do browse. Hopefully the problem is solved. Okay. So, okay, so I think we are set. We just need to change the DC to 2. And I think I can manage. Okay, so I think I have changed the code now, and let's run it once again to see if everything is fine or not. I hope everything should be okay. This is strangely taking a little bit more time. Yeah, something in that direction. So essentially, I think it can be even beautified if I just remove. So I think there was a limitation in the x-axis, so it should do. I think, yeah, something in that direction. So, I mean, obviously you can see that there is a huge portion which is non-significant, but you see if you turn this around, it becomes a volcano. So it's an inverted volcano. So the down regulation is with the blue dots since it is here, and the few up regulation is in this side, which is in the red dots. Yeah. So I think I would sum up quickly what we have done. I will go back to my presentation. That what we did so far, and just to give a summary. So today we did the entire RNS, at least one part of the RNSec, where we first checked the quality check. We then had certain introductions with the RStudio, and then we generated the count matrix, and followed by MDS plot generation. Then we found the various differentially expressed genes. We did some visualization. I'm afraid heat maps won't be possible, but since you guys have the code and feel free to walk around, I think there is even an online website called Heat Mapper that can also do heat maps. And I will give you little bit to the end, little intro to the end part, that's with the big data. So that's what also I do sometimes. That means that if you have a very complex layer of omics data, let it be transcriptomics data or maybe other omics, then you can use these modern day machine learning algorithms, which include certain clustering algorithms. And of course, at the end of all, we have deep learning. So that's neural network, and then which has many layers which interconnects to one another, and then it gives ultimately an output. Then it's much more. It has much more use in clinical applications, so to say. And I think if you do clusters, so clusters means finding groups where similar data fit together, and then you can also take a look there how it fits, and of course what kind of pathways are involved in various clusters and how they impact your data at the end of the day. So that would be basically it with my talk for this session that was beautifully organized. And with that, I think I am done with my presentation, and I would now invite for any kind of questions. Thank you very much. Ma'am, I think you are muted.
Oh, thank you, Sabarna, for such a wonderful session. I'm overwhelmed. But I'm sure our participants are benefited, and at least we know that R can be used in various ways. There are several tools that are available, and so many other things. The
Yeah,
volcano plot, it was so
yeah, yeah.
nice. Upward
Yes,
volcano.
yes, yes, yes.
So it
So,
was wonderful having you here and delivering the session.
thank you,
And
ma'am.
I'm sure that our participants will repeat this session after looking at the recording. So thank you very much. It was
It was my
pleasure
pleasure, ma'am.
interacting with you.
It was my pleasure, ma'am, as well.
Thank you.
Yeah. Thank you everyone for participating, and I think if there are no further questions, I think I wish everyone present all the best. And I mean, my email ID is here around, so if somebody does transcriptomics at some point, feel free to ask me questions also. So I am also available by email.
Thank you, Sabarna. I also want to thank
Thank you.
you for this wonderful, hands-on session on R, which is, I think, the basis of all the most of the bioinformatic work. So, giving an
Hmm.
insight into a very, very deep insight into that. Thank you so much for that.
It was really a pleasure, ma'am, to do this, and I think I think I somehow I enjoy doing art. So yeah, so it was also really my pleasure to be here.
Thank you, Shabarna. Thank you so much. And it's always a delight to have our students, come as an expert. So it's really an achievement for teachers is that only.
Yes, ma'am. So I think with that, I will say goodbye to everyone, and I think at some point we can meet, maybe offline as well. Yeah,
Sure, we
All
will.
right.
Sure, Sabarna.
yeah,
Let us know whenever you are here in India. So we will
yeah,
surely like to meet.
yeah. Yes, ma'am. Okay, ma'am. I will
All
wish everyone a very nice evening ahead. So, bye.
Bye bye. Take care.
Bye. Bye. Bye.
Bye, Shabarna. Take care.